Hello, I'm Rebecca Folkerth. I'm a neuropathologist in Boston, and I'll be presenting neuropathology of the peripheral nervous system and muscle, an update. Notable findings of the past decade include a greater understanding of chronic inflammatory demyelinating polyneuropathies, and among many different things going on in skeletal muscle, I'd like to talk with you about molecular profiling of inflammatory myopathies, mutations in sarcolemmal anchoring structures, such as the dystrophinopathies, in particular the glycosylation defects which affect brain development, and mutations in sarcoplasmic proteins leading to the myofibrillar myopathies. CIDP is a disorder involving the motor system and sensory system. It can be separated into several forms clinically based on whether there is symmetric involvement of motor fibers and based upon the findings of nerve conduction studies. Pathologically, these are all due to essentially the same process, just depends on which nerves are involved, in which the myelin sheath, shown here, begins to fragment and exposes the underlying nerve fiber or axon. And this is thought to be mediated by autoantibodies and by macrophages, which then ingest the cell debris. Histologically, in neuropathology, this is the type of section that we see. This is a longitudinal section of a peripheral nerve, a sural nerve biopsy, with a trichrome stain. And a trichrome will stain the collagen green and the myelin sheath red. And a normal myelin sheath has this bubbly appearance, fine bubbly appearance, whereas these larger, irregular accumulations of material indicate myelin breakdown. It's not possible on the basis of this particular preparation to tell if the axons are affected or preserved, and for that we will go to ultrastructural studies. But in addition to the myelin breakdown that's shown here, one will also see macrophages or inflammatory cells around small blood vessels within the nerve, as shown here. If one does a nerve tease preparation in which individual nerve fibers are fixed in osmium tetroxide and then teased out onto a glass slide, one can see the segments of demyelination here. Here's a normal myelin sheath, here's an internode, and here's a hypomyelinated segment of the axon. Here's another one. And notice also that this internode is shortened between the nodes, and this is a consequence of demyelination and remyelination. Here are some beaded up fragments of myelin similar to what was seen in the trichrome stain, those red accumulations that were present. And here's a very thinly myelinated fiber here. So this is a highly abnormal nerve preparation. So if we look at an ultrastructural preparation of the nerve, here we have a cross section showing the axon, the myelin sheath. This is the basal lamina of the Schwann cell, and we see all of this myelin breakdown material within the cytoplasm of the Schwann cell. Here's also a smaller myelinated fiber, and we also see debris present in adjacent cells, indicating some chronicity. This is what one sees if the process continues on for some weeks or months. This is a so-called onion bulb, where we have a remyelinated fiber here. Here's the axon and the myelin sheath, but around it are a number of extra or supernumerary Schwann cells that divide as a response to the injury process and form these concentric laminations around the axon. Criteria for making the pathologic diagnosis of CIDP have been recently defined. These include mandatory criteria such as a nerve biopsy showing unequivocal evidence of demyelination and remyelination, such as we've seen in the last couple of slides. And this can be either by electron microscopy, looking at more than 5 fibers in a nerve, or by teased fiber studies. and there are criteria here for the amount of abnormality that must be seen in order to have certainty of the diagnosis. There are supportive criteria as well, being edema, mononuclear cell infiltration, onion bulb formation, and a prominent variation in degree of demyelination between fascicles. This is important as it's definitely not a uniform process among all of the nerve fascicles, which might suggest something like ischemia if it were seen. Exclusionary criteria are the presence of vasculitis, swollen axons with neurofilament accumulation, amyloid deposits, and so on, or evidence of other types of processes which could explain the abnormalities in myelin. What has been understood recently is that the components of the Schwann cell axonal contact region here in the node of Ranvier are the targets for the inflammatory process. Here we've got a stylized axon with the node here with the folds of myelin coming around, the adaxonal membrane. Notice that in the nodes are where the sodium channels are aggregated, whereas the potassium channels are in the internodal regions. In the regions of the node, there are a number of different proteins, neurofacin 155, for example, and gliomedin and neurofacin 186. These are localized specifically in these regions and actually connect into the cytoskeletal protein system, which is not depicted in this drawing, but is present in the axon that lends stability to the structure. So antibodies can affect these particular protein components and cause them to detach and disrupt, and therefore cause degeneration and breakdown of the myelin. We see also here proteins in the myelin sheath, specifically P0, and antibodies and T-cell response has been also identified to myelin proteins and also to these specific ion channels. So there are a number of different ways in which these the myelination can be affected in the axon. So this is not just one disease. And for example, Guillain-Barré syndrome or AIDP, acute inflammatory demyelinating polyneuropathy, one can also find ganglioside antibodies in the nodal region. And this is an example, a diagram showing the effect of the antibodies on these ganglioside proteins here, the GM1 ganglioside. And these are derived from B cells, either in the regional lymph nodes, and it's thought that perhaps these may originate from Peyer's patches in the gut. It's also possible that these could be triggered by some sort of viral infection. So to summarize some of these recent advances in understanding CIDP, many pathways exist to disrupt the axonal and Schwann cell membrane molecules needed for the integrity of the myelin sheath. And so this is probably not a single entity, even though it appears under the microscope very similar and clinically has some shared features. The protein components are presumed antigenic targets, and there may be a viral initiator with molecular mimicry underlying the susceptibility of these particular targets. And eventually treatment, which is currently just supportive or involved in immune suppression, will need to be directed to the individual mechanisms once they're better understood. Now I'd like to talk about idiopathic inflammatory myopathies, which I'm sure everyone is familiar with. And these are dermatomyositis, polymyositis, autoimmune necrotizing myopathy, and inclusion body myositis. Here's the typical periorbital edema of a patient with dermatomyositis. There's also a bit of a heliotrope rash here. This is seen in a child who looks particularly miserable. This is a very uncomfortable disease for them. And then these are the papules seen on the extensor surface of the hands. Also, one can see the dilated capillary loops and even ulceration in the nail beds. The muscle biopsy characteristically shows perifascicular atrophy, which is made up of small degenerating and regenerating fibers. There's disrupted oxidative enzyme staining there and microvacuolation. And it's often been thought that this is caused by ischemia, although it's not clear that that's the case. But there are other features that suggest ischemia, such as scattered necrotic fibers and wedge-shaped microinfarcts. Inflammation tends to be perivascular and is primarily located in the perimysium and is composed of macrophages, B cells, and CD4 positive dendritic and T helper cells. There should be no invasion of non-necrotic muscle fibers by inflammatory cells in this disorder, in contradistinction to polymyositis, which I'll show you in a minute. An early histologic feature is a reduction in the capillary density, and some investigators have wondered if this is a cause or an effect of the disease. One thing that has been noted is the presence of membrane attack complex, or complement 5B through 5-9, protein deposition on or around small blood vessels. Again, it's not clear whether this initiates the process and then there's destruction of the muscle, or whether destruction of the muscle results in accumulation of these inflammatory mediators. There may also be complement and IgM and IgG deposited in these areas, and electron microscopy shows a typical endothelial hyperplasia, microvacuoles, and cytoplasmic inclusions in the muscles. This is a typical appearance of perifascicular atrophy. So here's the edge of a muscle fascicle. These are more normal appearing fibers here up in the corner. And you can see these are much smaller, have a bluish cytoplasmic appearance, which indicates the presence of increased RNA and protein synthesis for regeneration. And also the nuclei are more centralized. These are the capillaries that are in the between the fascicles, showing a mononuclear cell infiltration, and even sometimes one may see frank necrosis of these vessels, which leads one to believe it could be a primary vasculopathy. This is an immunostain for membrane attack complex, and I'm sorry this is a little bit faint, but you can see that the capillaries in between the myofibers look like they have some deposition, and there's also some staining along the sarcolemma in some cases. The pathogenesis is thought to be due to plasmacytoid dendritic cell infiltration and upregulation of type 1 interferon-inducible genes and proteins. In fact, PDCs are interferon alpha and beta factories churning out this cytokine. They're also called natural interferon-producing cells, and it's hypothesized that these have a direct toxic effect to the small blood vessels and the muscle fibers. The trigger is unknown. Again, as mentioned before, it could be a viral infection. The plasmacytoid dendritic cells are seen here highlighted by a BDCA2 immunostain, and you can see they have these processes, and dendritic cells are involved in presentation of antigen in certain circumstances, but they should never be to this degree upregulated within a muscle, and so this is pathologic. And in fact, these cells, although this is not a double labeling experiment, one can see that cells with the morphology of BDCA2 positive plasmacytoid dendritic cells also express interferon alpha, so that these are considered the origin of this particular cytokine, which is the one causing the damage. If one looks in the peripheral blood of patients with dermatomyositis, and this is a heat map showing not just those with dermatomyositis, but also those with polymyositis, inclusion body myositis, and necrotizing myopathy, one can see that there is a distinct upregulation of genes in the interferon gamma pathway, and that this signature is present in the blood of patients with dermatomyositis versus those with polymyositis. So that this is a very different mechanism than one sees with the other inflammatory myopathies. One can also track the response to treatment by looking at these interferon alpha inducible genes in the peripheral blood, and this is showing initially and then after treatment one can see that these decrease dramatically, except in the case of a refractory situation in which there is no decrease, so that this is a means of tracking progress of the disease in response to treatment. The prognosis is variable. but overall fairly good in adults. It used to be a terrible fatal disease in children, which is not so much the case. However, there are poor prognostic features which include the presence of an underlying malignancy, obviously. Those older patients do tend to do a bit worse. Interstitial lung disease, cardiac disease related to the inflammatory manifestations, and late or previous inadequate treatment. To contrast dermatomyositis with polymyositis, we see a different picture neuropathologically as well as clinically. The muscle biopsy shows variability in fiber size with scattered necrotic and regenerating fibers and endomesial inflammation, and the sine qua non is the invasion of non-necrotic fibers by inflammatory cells which express MHC1 antigen. In some textbooks and practice, this is not necessarily seen. It also depends on how adequate the biopsy is. One may just see endomesial and perivascular inflammatory cells. However, this is, I think, an important feature to look for. The inflammatory cells include macrophages and CD8 positive cytotoxic T cells. This is in contrast to dermatomyositis, in which the infiltrating T cells were CD4 positive. There can be a T cell receptor repertoire which is increased in the endomesial compartment, and they can demonstrate an oligoclonal pattern of gene rearrangements and a restricted motif in the CDR3 region, suggesting an antigen-driven response. There's also an oligoclonal expansion of B cells and plasma cells in the muscle tissue. And in contrast to dermatomyositis, there are not the evidence of immune deposits present. Here is an H&E section of polymyositis showing the muscle fibers, which vary quite a bit in diameter. Some have central nuclei, and here is an intact, structurally intact fiber with incursion of inflammatory cells into it. So this is the invasion of a non-necrotic fiber, which is typical of polymyositis and extremely helpful in making the diagnosis. Using an immunostain to MHC1, we can see that these cells that are invading the myofiber are MHC1 positive. There's also some expression on the membranes and in the inflammatory cells in the endomesial space. Expression studies, as we were just looking at in dermatomyositis, also show some slight increase in type 1 interferon-regulated genes, but not upregulated so much as in dermatomyositis. And expression studies in the muscle tissue show an increased expression of immunoglobulin-related genes. This is of unclear significance, but it may be useful in identifying possible antigens in an experimental setting. So, pathogenetically, the histological and immunological studies suggest that it's a result of an HLA-restricted, antigen-specific cell-mediated immune response directed against muscle fibers. And while the etiology is unknown, again, viral infections have been postulated. There are no, at this point, conclusive findings which support this theory. Most polymyositis patients will do well with immunosuppressive treatment, and some suggest that, some retrospective studies suggest that it doesn't respond quite as well as dermatomyositis, but this again depends on the age of the patient and when they're treated, whether there's interstitial lung or cardiac disease, and whether anti-SRP or Jo1 antibodies are present. There are overlap syndromes, which can be the case in which dermatomyositis or polymyositis is associated with some other well-defined connective tissue disease, such as scleroderma, mixed connective tissue disease, SLE, and so on. These do not have definitive separating features histopathologically, and the clinical setting and presence of antibodies must be taken into account. Necrotizing myopathy is quite distinct from both polymyositis and dermatomyositis, although it can resemble it somewhat clinically and will often require a muscle biopsy for definitive diagnosis. The weakness tends to be proximal greater than distal and is often acute in onset with markedly elevated serum CKs. Again, it may be associated with cancer or a connective tissue disorder. Much more commonly, we see it in the setting of statin use. There are some associations with SRP antibodies and antibodies directed against HMG-CoA reductase, the latter, again, associated frequently with statin use. So this is a more of an antibody-mediated disorder. The typical appearance shows of the muscle biopsy shows scattered necrotic fibers with hardly any inflammatory cell infiltrate. This is aside from the myophagocytosis of necrotic fibers, which I'll show you in just a minute. One may see thickened pipe stem capillaries, but not the sort of necrotizing change that is present in dermatomyositis or inflammation. One can see MAC deposition on the sarcolemma of non-necrotic muscle fibers in the statin cases, and this particular disorder requires aggressive immunosuppressive treatment. The etiology, aside from those associated with statins, is unknown. This is a typical appearance of a skeletal muscle biopsy on trichrome of a necrotizing myopathy. We can see the muscle fibers here. Here is a fiber undergoing myophagocytosis with macrophages and necrosis of the cells in the cell material in the macrophages. We also see some aggregation of the non-contractile portions of the cell here, but otherwise there is no endomesial inflammation and the vessels are not involved. Inclusion body myositis is also very distinctive appearing. This is a patient with inclusion body myositis showing severe distal atrophy. On the muscle biopsy for inclusion body myositis, we see endomesial inflammation composed of CD8 positive T cells and macrophages, again invading non-necrotic muscle fibers. We can see expression of MHC1 antigen. We can see small groups of atrophic fibers, so that sometimes these cases look as if they have both neurogenic and myopathic features. The typical appearance and the pathognomonic appearance is the presence of rimmed vacuoles lined with granular material. And looking closely, one can also sometimes find eosinophilic cytoplasmic inclusions. Amyloid deposition has been described, and by electron microscopy, one can find the typical tubulofilamentous inclusions in the cytoplasm and sometimes in the nuclei. There may also be ragged red fibers, and this should not lead one to think of a mitochondrial disorder, because normally mitochondrial disorders have a very different clinical appearance and do not have the typical rimmed vacuoles and so on that are present in inclusion body myositis. This is an H&E section of an example of inclusion body myositis, showing numerous fibers which are small, some are large, so there's quite a bit of variability in fiber diameter. Here's some cleft-shaped vacuoles in the fibers, and this is incursion of inflammatory cells into a non-necrotic fiber. Some cells show very prominent granular material in the rimmed vacuoles, and these can be positive on trichrome stain, as seen here, showing very red granular material. These have a different appearance than the ragged red fibers, and instead have these clefts, which are not seen in mitochondrial myopathies. One may occasionally see Congo red positive apple green birefringent material. This is lipofuscin here, and this is amyloid. This, however, is the exception rather than the rule. By electron microscopy, the characteristic tubulofilamentous inclusions are seen here, which look a bit like a tangle of yarn, or spaghetti and meatballs is one thing that's I've heard said before. And in conjunction with the typical inclusions, there is the presence of extensive cell debris, including myelin figures, which indicate excessive cell turnover and injury. Here is another tubulofilamentous inclusion in between the sarcomeres here. Again, sort of skeins of yarn with little dots. And here is one present within the nucleus. This is chromatin here, and this is the nuclear envelope. And this is the subsarcolemma. This is the sarcomere here. The inflammatory cells in inclusion body myositis include CD3 positive T cells, seen here. CD3 is a pan T cell marker. And of these, most of them are CD8 T cells. One can also find BDCA1 positive dendritic cells, not so much BDCA2, but CA1 around these fibers and with some incursion into the intact fibers. Plasma cells can be present. This is a pan B cell marker, CD20, which picks up a small complement of B cells, but a specific plasma cell marker, CD138, is present extensively throug'out the fiber. Interestingly, a recent autoantibody has been discovered to cytosolic 5' nucleotidase 1A and T5C1A. This is a cytosolic enzyme that is most abundant in skeletal muscle. It catalyzes the nucleotide hydrolysis to nucleosides and is involved in a variety of functions, including the regulation of deoxynucleotide pools. So it's very important in DNA repair and regeneration, so that the presence of this autoantibody suggests that there may be a problem in DNA repair and metabolism. These are common and highly specific among muscle diseases and may provide a link between the dual processes of autoimmunity and myodegeneration in IBM. The reactivity by immunohistochemistry can be seen in perinuclear regions and the rimmed vacuoles, localizing to the areas of myonuclear degeneration. Blood testing is feasible and should improve early and reliable diagnosis of IBM, but I'm not sure that this is translated completely into the clinic yet. What it looks like histologically is the presence here. This is immunofluorescence with a red signal here showing a accumulation around the nucleus, which is here in blue. Again, this is the presence of the enzyme, and again, it should be configured close to the nucleus based on its purported function. And here you can see, using a typical IHC with a brown chromogen, the presence in some of these involved fibers. The T cell receptor repertoire is expanded and shows an oligoclonal pattern and heterogeneity in the CDR3 domain, and this suggests a possible T cell response to a superantigen. However, there is also some oligoclonal expansion of B cells and plasma cells, and an increased incidence of monoclonal antibodies in the serum. In particular, there's one that's directed against an unknown 43 kilodalton muscle protein in about half of patients. The pathogenesis, in spite of the evidence presented so far, is not completely clear. It does appear to be separate from the other inflammatory myopathies, but it's not clear whether the injury occurs to the muscle and then there are secondary changes, or the muscle is a bystander to another systemic problem. A potential clue is the finding of the abnormal accumulation of DNA binding proteins in the sarcoplasm of IBM muscle fibers. This is the same protein that's been reported in the neurons of sporadic ALS, non-superoxide dismutase familial ALS, and some frontotemporal dementias. These are the TDP43 inclusions. They are normally present in the nucleus of normal cells, but are aberrantly expressed in the cytoplasm of affected cells here. This is a corresponding H&E section. And so the affected cells show this accumulation of TDP43, whereas the normal cells here only show expression in the nucleus. Again, immunostaining with a fluorescent antibody to TDP43 here, using a red fluorophore, shows an angulated fiber with accumulation of the protein in the cytoplasm, whereas the nucleus, here shown in blue, does not contain any of the protein. In contrast, the adjacent normal cells show a purple signal, which is a merger of the blue nuclear signal and the TDP protein, showing a normal distribution. So that, again, this is a marker for the sick fiber, the affected fiber in IBM. And if one counts the number of affected myofibers using this marker, it's actually much it's a much better marker, for example, than even rimmed vacuoles, far better than Congo red fluorescence or other markers that one can see. So this is extremely useful and is in place in some centers as an immunohistochemical stain, as an adjunct to confirm the diagnosis of inclusion body myositis. In recent years, molecular profiling of inflammatory myopathies has been useful in classifying these disorders. As we've seen earlier in dermatomyositis, these allow separation based on gene expression of different classes of inflammatory myopathies. Here, circled is the earlier group that we saw of dermatomyositis. This is polymyositis, polymyositis here, and inclusion body and necrotizing and normal are all quite different. So to summarize, the idiopathic inflammatory myopathies are clinically, histologically, and pathogenetically distinct. Dermatomyositis is an immune-mediated disorder, possibly due to a direct toxic effect of type 1 interferons on small blood vessels and muscle fibers, whereas polymyositis is a T-cell-mediated disorder directed against muscle fibers. For the necrotizing myopathy and inclusion body myopathies, the pathogenesis is less well understood, but suggests immune and possibly neurodegenerative aspects, respectively. Now I'd like to move on to discuss a couple of entities among the muscular dystrophies. This is a huge family of entities, including the Duchenne's, limb-girdle, and so on. I'm going to discuss a subset of congenital muscular dystrophies, which also involve the brain, and myofibrillar myopathies. Myofibrillar myopathies can also be classified under the distal dystrophies, and we'll go over the details of this in just a minute. I will not be discussing these others in the interest of time. Congenital muscular dystrophies with glycosylation defects and abnormal brain development, or cobblestone-less encephaly, involve a group of disorders of which Walker-Warburg syndrome is the prototype. There's also muscle-eye brain disease and Fukuyama dystrophy. And these are characterized by a very interesting abnormality in post-translational modification of genetically structurally normal dystroglycan. So the protein itself, the gene for dystroglycan, is intact. This is in contrast to the limb-girdle and Duchenne's type dystrophies. But because there is not a functional glycosylation of the protein, there is decreased binding to the extracellular matrix proteins, leading to a myopathy and also abnormal brain developments. A handful of known gene mutations account for the majority of these. For the glycosyl transferase mutations, these are autosomal recessive and include these disorders here. And the O manacylation disorders are the laminin binding protein disorders, which include these disorders here. If one looks at a set of fetuses with cobblestone-less encephaly as a disorder in common, we can see that there is a wide variety of abnormalities that can give rise to this phenotype. As I said, most of these have a known gene mutation, but there are some others, a good proportion, that do not. And of those that do, these are the POMT1, T2, and so on, fukutin-related protein deficiencies that result in a similar appearance of the brain. What this looks like is a irregular cobblestone surface. This has also been called type 2-less encephaly, and it can have variable regions of involvement depending on the type. And what gives it this cobblestone appearance is that there is an abnormal growth of neurons and glia outside the normal glia limitans. And here you can see it sort of flowing over the surface of the brain. Here is the the familial kindred analysis showing that this is actually a consanguineous pairing here, and these are as an autosomal recessive example of cobblestone lissencephaly. This has a typical appearance radiographically as well, and one can see that there is a disorder of glycosylation. So here in the patient versus the control, we have fibroblasts and myoblasts, which show normal glycosylation in the controls and absent glycosylation in the patients. And this gives rise to a typical dystrophic appearing muscle with wide variability in fiber diameter, extensive endomesial fibrosis, and degeneration and regeneration of fibers. And if one applies immunofluorescent antibodies to this muscle, you can see that in the Walker-Warburg syndrome, which is what this is an example of here, that there is normal dystrophin, but there is an absence of normal staining for the alpha dystroglycan in these cases, the Walker-Warburg cases, whereas beta-dystroglycan and myrosin are unaffected. How does this result in abnormalities of the brain as well as abnormalities of the muscle? It's similar to the dystrophic muscle, which, as you know, is due to a lack of anchoring of the skeletal muscle apparatus to the basement membrane because of the lack of dystrophin. In the brain, it's similar because of the abnormal dystroglycan adhesion, which normally allows the neuron to migrate up and stop in the appropriate place in the cortical plate. The absence of this results in gaps in the glia limitans so that the neurons, as they migrate up, are not prevented from migrating out into the subarachnoid space and causing this coating over the surface of the brain, or so-called type 2 lissencephaly or cobblestone lissencephaly. So this is an example of a protein that has double duty both in the muscle and in the brain in normal development. In contrast, myofibrillar myopathies have an unusual structural accumulation within the myofiber itself. These are a clinically and genetically heterogeneous group of disorders and show a pathologic finding of myofibrillar disruption on EM and excessive desmin accumulation in muscle fibers. Sometimes these are called desmin storage myopathies. In fact, they have a wide range of other names, which I'll show you in a minute. And because desmin is not the only protein that accumulates, the term myofibrillar myopathy was suggested by Engel and is probably a better term. Here are the many other names by which it's been known over the years, and again, this is an indication that this is not a single disorder, but has many underlying abnormalities which can lead to a similar phenotype. Clinically, there's a wide spectrum of phenotypes, but most patients tend to develop weakness in early to mid-adulthood. Weakness may be noticeable in infancy or present later in adulthood, though, as well. It can be proximal, distal, or generalized, and some patients have an FSH or scapuloperoneal type of distribution. Many have cardiomyopathy as well, and this may result in cardiac arrhythmias and congestive heart failure, which may be some of the predominant features of the disease. In fact, patients with cardiac arrhythmias and CHF who are a fairly young age and may have relatively clinically unrecognized weakness should be considered for testing for abnormalities of myofibrillar myopathies. Some of them may even require pacemaker insertion or cardiac transplantation. They can also have severe respiratory muscle involvement, although this is relatively rare. In addition, smooth muscle can be involved and lead to intestinal pseudoobstructions. They may have moderately elevated serum CKs, and the EMGs are often irritable with myotonic or pseudomyotonic discharges. And they, on EKG, may have conduction defects or arrhythmias. And echoes may show dilated or hypertrophic cardiomyopathies. Microscopically, what we see on H&E is, again, the hallmark of an abnormal muscle, quite a bit of variability in myofiber diameter. Here's a very wide fiber and a very angulated small fiber here. And one can see an abnormal aggregation of material right in the center of the fiber. Here it's scattered throughout central nuclei, and this has a very hyalinized appearance, pink or glassy appearance, so-called. Other fibers may have more larger, more irregular such inclusions, or some more fuzzy type changes in the edge. And again, they can have nonspecific myopathic features of fiber splitting, centralized nuclei, endomesial fibrosis, and so on. On trichrome, these inclusions have a more reddish purplish appearance, can be very smudgy, and may sometimes be seen in a minority of fibers. For example, a lot of these fibers are still relatively normal looking. In addition to these hyalinized aggregates, again, one can see sort of fuzzier types of inclusions as well. And on the NADH tetrazolium reductase stain, we can see an absence of oxidative activity within this hyaline inclusion. That's because there are no mitochondria in between these aggregations. And ultrastructurally, these correspond to dense aggregates of filamentous material. Sometimes one will see also nonspecific degenerative changes such as myelin figures and vacuoles. And the hyaline lesions have this distinctive appearance, sometimes with a little bit of a halo around them. So the etiologies are variable, and many different mutations can give rise to a similar appearance. They can be X-linked, and these can be mutations in FHL1. They can be autosomal dominant and affect genes coding for myotilin and some of the other abnormalities listed here. And they can be autosomal recessive with mutations in the selenoprotein N gene associated with Mallory body-like inclusions. Those would be the hyaline type of inclusions. So in summary, congenital muscular dystrophies are expanded to include disorders of glycosylation and otherwise genetically normal structural proteins. So these are different from the structural protein disorders. And these are part of the cytoskeleton, which are actually distinct from the dystrophin complex. Trends to watch. Immunohistochemical or other methods for detection of expressed proteins specific to disease etiologies may mean that there's less emphasis on electron microscopy in the future, although for patients with a relatively uncharacterized disorder, this is still a useful test. Molecular diagnostics may be performed with serum markers and genetic studies to supplement or even replace nerve and muscle biopsy in some cases. And I'd like to acknowledge the help of Dr. Anthony Amato, Dr. Stephen Moore, and Dr. Umberto De Girolami. Thank you very much.